In this video, I'll talk through the 2024 National 5 Biology paper and explain how I would approach each question. Starting off in the multiple choice question, we have 4 different cell structures. We are asked about producing enzymes. First thing we need to know is that enzymes are proteins, and proteins are formed in the ribosome. Mitochondria is the site of aerobic respiration. The nucleus contains the genetic information, also controls the cell activities, and the cell membrane controls what enters and exits the cell. Okay, so they tricked me at first. I was thinking it was something to do with the magnification, but what they're just asking is what's the average length of the cells shown? Well, if it's 1200 micrometers across from left to right, we have got 1, 2, 3, 4, 5 cells across. So if 5 cells is 1200, then to get what one cell would be, we divide by 5. You do the same on both sides. So 1200 divided by 5 gives you 240 micrometers as our answer. In this question, we're talking about the difference between passive and active transport. Mitochondria are involved in aerobic respiration, and that releases energy. So in this question, we're looking for active transport, which goes from an area of low concentration to an area of high concentration. In A, we have equal concentrations inside and outside of the cell, so there'll be no movement at all. B, we've got diffusion as we're going from high outside to low inside. That's not our answer. In the next one, we're moving from high concentration to low concentration, so C would be diffusion as well, which must mean D is the correct answer because it's the only one that's moving from a low concentration to high concentration, and that would be active transport. Next one, we are looking at a section of DNA. DNA has A, E, G, and C, adenine, thymine, guanine, and cytosine. We're told that 32% is guanine. G and C are complementary bases, which means we must have the same percentage for the cytosine as well. Our total is going to need to be 100%. So if we take away the 64, which is those two added together, that leaves us with 36%. And that's a combination of the A's and the T's. And because they're complementary, they both have to be the same. So they would both have to be 18%. And then we can find out what 18%, well, 18 over 100 is 18%, times by 3400. And that gives us 1152 and the answer of A. Another approach you could do is you could deal with the 32% straight away. So if we find out what 32% of the 6400 is, this means that our G's and C's both have to be 2048. And then this time, if we do it 6400, take away the 4096, and that means that 2304 is the A plus T, and that means that A is going to be half of that, which should give us the same answer, 1152. I prefer to keep it in the percentage, just keeps the numbers more simplified at the beginning, but either way would work. We both get the same answer, and that gives us A for that one. Going on to question 5, we're asked about the product of concentration during a degradation. In an enzyme reaction, which this is talking about, we start off with our substrate, and that is changed into the product. The product has been formed, which means that over time, we will be making more of the product as we go. The answer has to be C in that case. A kind of looks like one of those ones about the optimum kind of values, which isn't what we're talking about. The substrate's concentration would go down over time, which would be that one. And then D, nothing's happening, so maybe that would be like the enzyme doesn't get used up. So that would stay there, but the answer is C here. In genetic engineering, there's 2 sets of things that have to happen. One thing that has to happen is we have to get our desired gene extracted from the chromosome. And we also need to have the plasmid being extracted and then altered to allow the gene to be put into the plasmid to modify it to be able to then go in and grow it. With these orders here, the fact that we've got one of them that says that the plasmid needs to be extracted from the bacterial cell, well, you can't cut open the plasmid until you've extracted the plasmid. So 3 has to go between, so 3 has to happen before 2. We're going to extract the gene that we want, and we're going to insert it into the plasmid. Means the answer is D. With these types of questions, you're best looking at them as an entirety rather than separate statements. So if one's talking about changing something about the plasmid, well, you have to get the plasmid out first. That then modified plasmid gets inserted into a host cell, which is then grown to make lots of the protein that we're trying to make. In a question about reliability, I would be thinking about repeats. Now, in A, we're looking to extend the temperatures we're investigating. That's not going to impact the reliability. It might make our results more helpful moving forward. If we're testing B, we are setting up 10 solutions at each temperature. That's repeating them at each temperature. So that's going to be my answer I'm going to pick. Using the same concentration makes the results more valid. We've got a fair test so we can see exactly what the effect of the temperature is. And again, using the same types of cells, that again is making it a fair test and making it valid. Going through, B would be the correct answer for that one. On to number 8. When we're looking at a control experiment, we want to repeat the experiment but taking away the variable we are investigating. So we're investigating respiration in maggots. So looking for one that hasn't got the maggots in it. Looking at A, what's the difference with A? So the difference is where the maggots are. And so in A, they've just swapped around the maggots and the chemical to absorb carbon dioxide. So that's not going to be our answer there. Next one, the beads are where the maggots were. Everything else looks the same. That looks quite helpful there. And C, yes, we have the glass beads, but we don't have any of the chemical to absorb carbon dioxide, which isn't what we're asked to do. And the last one, it's the same setup apart from no chemical to absorb carbon dioxide, and it's not going to be that one there. So looking at the maggots, so repeat it either with a replacement for the maggots, or sometimes they might even do it with dead maggots to see that the maggots have to be alive to carry out respiration. On to number 9. We're talking about spindle fibres, and it stops spindle fibres. So which of the stages would still occur when this colchicine is present?
Colchicine.
So chromosomes moving to opposite poles of the cell is caused by the spindle fibres contracting and pulling them. It's nothing with that. B, pulling them apart by the spindle fibres, not that. Shortening and thickening would be able to happen. That happens earlier before they start getting pulled apart. 2 diploid cells, well, if you can't pull them apart because you don't have spindle fibres, then you can't make the 2 identical daughter cells, so that wouldn't be true. So C would be the correct answer in that one. Next one, we're talking about the cerebrum. That is for memory and conscious thoughts. So remembering, that would be memory. That's that. The heart rate is the medulla. Coordination is the cerebellum. And that's balance as well. Coordination and balancing. That must mean that A is our correct answer there. Next one, we're talking about hormones and their production site and its target. Okay, when dealing with blood sugar levels, insulin and glucagon are the hormones. Glucose and glycogen are the chemicals that are converted between one another due to insulin and glucagon. Those are both made in the pancreas. So insulin's not made in the pancreas, which means D has to be correct. It is targeting the liver, which stimulates the release of glycogen and breakdown to glucose to increase the blood sugar levels back to normal. Insulin would encourage the storing of glucose as glycogen to bring back down the blood glucose levels. Number 12, we're looking at a conclusion for these results. We just need to take our time working through. Reaction time always increases with age for both males and females. Let's look at the first set. Well, we've got it for males going down and then back up, so it's not always going down. The females, it's always going up, so it's not going to be A. B, again, because of the males, having a wee dip and then back up, that will not be true. Females have a lower reaction time than males in each age group. Now, rather than looking at the actual numbers, I'll just run a line across and go, okay, so males are lower, males are lower, males are lower, males are lower. So that is, the females isn't true, they have not got a lower one, so C is incorrect, but D is the other way around, and then that would have to be our correct answer for number 12. Just take your time, make sure you're looking at the key, seeing which age group. They might have changed it up and said, oh, this age group has this, whatever. Just take your time and then that should go smoothly, hopefully. And this one, on the plants, we have the female part, which is the ovary, not the ovule. Ovule is what's found in the ovary, and it's just talking about the structure. It'd be a bit tricky, they might have asked us about what's made in structure G. This one's just in the name of structure G, so ovary. And the cells that are produced, the ovules that are produced, have half the information, which is why it joins on with the pollen to form the new plant. And yeah, that'd have to be haploid as well. So the gametes produced are haploid, half the information that's needed for the new individual. Number 14, we're talking about types of variation. Presence of absence of those. Okay, well, horns, you've got limited number of options, either present or absent. That means that's going to be a discrete variation. That means A can't be right because that says continuous. Height is continuous, not discrete, so that means it can't be C. For discrete variation, that's controlled by a single gene because it's less complicated options for it, in this case present or absent. Whereas in height, you've got a wider range of options for heights, and that is controlled by more than one gene, making it a polygenic inheritance, meaning B is our correct answer there. The stoma are the openings at the bottom of the plant. That's going to be your lower epidermis. Next one's about transpiration. I would think of it as drying clothes, either outside or inside your house. When it comes to humidity, it's how much moisture is in the air. So this would be, the air would be damper versus drier. If we want to have a decrease, so less water lost, or if you have increase in humidity, then the air already has water in it, so it's not going to absorb as much water from the leaf. So that would be one of our options. And then increasing the surface area, this means you flatten out the clothes, they will dry better. So increase in surface area would cause an increase in water lost, not a decrease. Decrease in wind speed, well, the wind helps move the air along and brings in dry air to absorb more moisture. So having less of a wind means it takes longer to dry the clothes / to lose water from the leaf, and D would be my correct answer there. On to 17, we're looking to do a prediction. As the pH is decreasing, we are decreasing the percentage bound. So it was 0.1 of a difference, made it go down by 4.5. Then by 0.2 of a difference, that's went down by 9. It's looking like every 0.1 pH, we're losing 4.5% oxygen. So at this point here, we are going down by 0.1. So my prediction would be that it will go down by it will go down by 4.5. That'll give you 55.5, which gives us answer C. So there is a trend going all the way down. Again, they might even try to trick us by having the first jump 0.1 and the second jump 0.2. But as we can see, the jump has doubled, 0.1 to 0.2. So has the difference in percentage, gone from 4.5 to 9.0. So that would fit in nicely. Next one, we're talking about the villi. A couple of things that might help you. So blood capillaries. From other topics, we're talking about blood glucose levels. So capillaries to the glucose and amino acids. Now the other type of structure we've got are the lacteals, and they deal with fatty acids and glycerol. So once you've learned them, what you can look at is you can look at the substances and look, well, fatty acids and glucose isn't a correct combination of substances. Amino acid and glycerol isn't a correct combination of substances. Amino acids and glucose is, but that's for the capillaries, not for the lacteals. And then lacteals take in the fatty acids and glycerol for that. So first step is knowing that those are the correct combinations. That takes it into C and D. And then you will have to try and remember that lacteals deal with the fatty acids and glycerol to get the correct answer there. But at least you can kind of narrow it down a part. Number 19, we're talking about a food web. And we're looking at what is true about these things. So we've got an owl and we've got a stoat. Now, they are an intra-specific competition. So is there anything that they're both going for? Well, they're both eating the wood mouse, sorry. So then they are in competition. Intra-specific means the same species, however. So they are in competition, but there's not same species. For the next one, okay, with B, we're talking about the vole and the shrew over there. Now, they are both eaten by the owl, but they're not eating the same thing. So they're not in competition full stop. If they were in competition, yes, it would be in pair. It's not, so it's not. Then the next one, ladybird is a predator and a prey. So the ladybird is eating the green fly, predator tick, being eaten by the shrew, prey trick. That looks like the correct thing there. Producers carry out photosynthesis. They will be bottom of the food chain. That would be some sort of tree or a bush, not an insect. That one's incorrect, so C is the answer there. In this question, we're looking at the difference between the populations. So we're on the maximum difference between the populations. So we want to go through year on year. Now, one thing you can do, you can look at the numbers, or we can just look at either how many boxes on this wee thing, or on my computer I can do the actual lengths between them. So we're at 6. Now, you might be able to sort of eyeball ones that look quite big. So we're getting a bit bigger. This looks like it could be a decent contender. We've seen them, we've got that 47. It's getting a smaller difference. We're starting to get bigger. Still smaller. And it looks like we now have a winner further up to here. So we're looking at that point there, and now we need to look at the values that they correspond to. So our predator, which is our full line, that was at 10,000. And just make sure we're always checking what the units are, because that again could maybe throw us off. 10,000, and then up the top, we're along at here. This has got every box must be 2. So ways of checking it, you can just try out numbers. So if you tried 1, 61, 62, it doesn't work. 62, 64, 66, 68, 70 does work. So 66,000. Another option is if you look at from here to here, we've got a difference of 10, and that's for 5 boxes. We'll divide them both by 5, means that every box has to be 2. So you can do it just by trying out numbers. Some things are quite nice, some things are a wee bit horrible, but we get to there. Difference between them, take them away from one another, gives you, what have I done? Okay, so I got tricked by that. My answer would have been 56, which isn't an option, thankfully. Otherwise, I probably would have lost that mark. They're asking us, rereading it, they're actually asking us about, look at the maximum for the prey, first of all. So let's go here. That would be 72. So you're looking at their individual maximums, not when their difference between them is the maximum, which is what I thought. Just what is their maximums. So we've got 72,000 for this one. And then this is the point where the predators are at the highest. And that would be 56,000. And when you take those away from one another, that'll give you 16,000 as your answer. So I definitely got thrown by the way that was worded. And each population was at its maximum. So that last bit's the bit kind of guides us a bit better for that. So look at each of the maximums and then put them together. But that I can very much understand people getting thrown off by that. On to the next one. Let's go on to this one, which is to do with a living factor. Moisture is to do with water, which isn't alive. Temperature is to do with heat, which isn't alive. Grazing is to do with animals eating the grass and the plants. That will be to do with living organisms. Then we just have a gas concentration, which is not living. They would all be abiotic or non-living factors. 22. We're looking at which leaves would have starch present. So for photosynthesis, we're going to need water. We're going to need carbon dioxide. And we're going to need chlorophyll in the chloroplasts to absorb the sunlight or light. Right, let's go through them then. So P, what's wrong with P? P has no carbon dioxide. P would not photosynthesize. Q has no light because it's covered in black paper. It will not photosynthesize. Leaf R has light. It is green, so that's the chlorophyll bit taken care of. Light can shine on it. It has access to the carbon dioxide in the air, and it has water coming from the roots in the soil. So that looks like it should just be R. Is the only one that does that. Next one. This question reminds me of an older question. Really try and throw us off a little bit. The number of eggs laid per female per day is what we need to check. So what are we told? We're told that they lay, how long they lay the eggs for, and how many eggs are laid for per laying period. Looking at the 20 degrees one, there was 108 eggs in 36 days. And we're asked about per day. So we need to divide both of them by 36 to see what one day would give. So 108 divided by 36 would give 3 eggs per day. And then we need to do the same for the other ones, which will be, oh, it's still 108 actually. So all of them 108 eggs. So divide it by 27 gives you 4. I'm pretty sure the next one would maybe be 6 or 5. Let's see, 108 in 18 days. That gives you 6 eggs per day. So as the temperature increases, the average number of eggs laid per day has to increase as well. Thinking another way, I hadn't clocked that they were all laying the same number of eggs. So if they're producing the same yield, how many eggs they're making, the one that takes less time to do that must be working faster per day. There must be more per day for that. My wee calculations does that as well, but maybe that's a way of avoiding doing as many calculations. A would be the correct answer there. When a fertilizer leaches, the algae start to grow rapidly, means they're increased in numbers, so the population would go up. This covers the surface of the river, and that will decrease the light that makes its way to the plants in the river. Both of them are decreased, so that's fine. Plants will start to die off because they can't photosynthesize, meaning bacteria will start to decompose them, and that will increase the number of bacteria. The bacteria use up oxygen as they're doing this, meaning that the levels go down, and A has to be the correct answer. So eutrophication is what this topic's dealing with there. Last one, we're talking about mercury building up within a food chain. We're asked about the small fishes and the mercury concentration in the small fishes. So the top of the food chain will have the highest concentration because it's eating lots of the little fish and absorbing all the mercury that they've eaten. That means that we have to have the second biggest value, which would be B for the small fish. So large fish, then small fish, aquatic insects, and plankton at the end would give you your answer of B for that one. Moving on to section 2, we're asked about cell structures. We have a single-celled organism. We have what's looking very much like a nucleus to me, site of genetic information. Function of a chloroplast is the site of photosynthesis. It contains chlorophyll, which can absorb the light to allow photosynthesis to happen. Why is this not a typical cell plant? So we're looking for something, a structure it doesn't have, that most of them would. A cell wall it does not have. No cell wall. Anything else? Did you just have a vacuole? Only need one of these, but or no vacuole. Or it tend to have, or should have, mitochondria. So no mitochondria. Next up is a little bit of a numbery question again. We've got the average body length of 50 micrometers. You can travel 1.3 micrometer, 1.3 body lengths in 1 second. How long would it travel in 30 seconds? Okay, well, if one body length equals 50 micrometers, well then 1.3 of them will equal 50 times 1.3. So 65 micrometers. And that's in 1 second. So if it's 65 in 1 second, then in 30 seconds, it'll be 30 times 65. And that gives 1950 micrometers as your answer. Could have done it the other way. If we wanted to, we could deal with the body lengths first. So if it's 1.3 body lengths in 1 second, it'll be 1.3 times 30. So that's how many body lengths it would make. One body length is 50. Times that by 50, you get the same answer. So in a bunch of these questions, it doesn't matter which order you do them in. As long as you're taking both of the things into account, you should still get to the same answer. In this one, we're asked to predict change in mass of 2 grams per 100 centimeters cubed. Now, where does that fit? Well, that fits slap bang in the middle of those two. So we are looking for, I would say, a number that's slap bang in the middle of them. There's 6 of a difference. So 3 more than 11 or 3 less than 17 would give you 14 as your answer. They might be quite lenient, it might get a bit of wiggle room somewhere in between them, but I would just try and aim straight in the middle. Yeah, that makes the most sense to me. Identify the salt solution where most of the cells would be turgid. So turgid is when they're plumped and puffed up and they have grown in size. And how's that going to happen? Oh, well, their size, it would get bigger. So 1 has the biggest increase in mass. So then that would be my answer for that one. If they were asked about plasmolysed, that's the one where they have shriveled up and lost mass. So 10 would be the one for that one. Okay, the told they all have a starting mass of 6 grams. Calculate the final mass of a piece of turnip in 8 grams per 100 centimeter cubed. Okay, so we have got start mass equals 6 grams. And at 8, they're saying that it's going to go down by 9%. If you're starting at 100% and you take away 9, we will be at 91% of our starting mass. Now I find the easiest to do percentages as well. 95 out of 100 is 91%. Times by 6. Back to the calculator. That'll give me 5.46 as the answer. Again, just for alternative options, if you worked out what 9% of 6 was, that's 0.54. It's lost that much mass, so 6 take away 0.54 does indeed give us the same answer. In this one, we're talking about the different types of respiration, aerobic and fermentation. Aerobic respiration is with oxygen. It produces carbon dioxide and water. So K has to be water. Glucose is first broken down into pyruvate before it then either goes and makes carbon dioxide and water and ATP or makes lactate, just depending on the presence of oxygen or not. State the number of ATP molecules that have been made from each molecule of glucose during fermentation. Well, in the initial stage, there's 2 ATP made. That's the only ATP that would be made in fermentation. Over here, we would get another 36 ATP for 38 in total. Each pyruvate is formed makes 18 when it goes through the left-hand aerobic part of it. So if it's asking you about just the left-hand part, it'd be 36 altogether. If it was going from glucose all the way through aerobic respiration, 38 is the answer for that. Had this earlier with the mitochondria. That's the site of aerobic respiration. We get from that. Why might they carry out fermentation, not aerobic? There would be no oxygen, less oxygen, or less oxygen present. There's not enough oxygen for aerobic. Aerobic needs oxygen. Next one, what type of reactions? Well, we have 2 reactions. We have synthesis and we have degradation. Here we are joining things up. That's going to be a synthesis. One way it might help you think about that is photosynthesis was making up sugar from carbon dioxide and water. So building stuff, making stuff. The substrate is at the start. The product is what you make. So it's going to be the glucose 1-phosphate. Okay, in this one, we're asked about time and how long it takes to make starch. Okay, first thing, why would you remove any starch that's already been made? Well, what B is in that? So that would prove, that's going to prove the starch has been made during the experiment. Whether there was starch being made or not made during this experiment, it would still show a positive result. 2 variables to make a valid experiment. So what things can make a difference on the experiment? So things to control, we'd want to control how much of each of the chemicals we're putting in, be that the glucose 1-phosphate or the potato extract, because having more of a substrate or more enzyme would make a difference to the experiment. Glucose 1-phosphate or the potato extract. The other thing you could control as well is the volumes of the solutions. Next one, explain how the results show that the enzyme is required for the reaction to occur. Well, in the control, there's nothing produced. So starch produced only when enzyme is present. Next one, we've got lactate building up in a long distance cyclist at different speeds. What was the lactose concentration at the start of the season when the cyclist was travelling at 40 kilometers per hour? So 40 kilometers per hour is here. Start tells you up here is the full line. That's the top of it here. I want to run that across to here. And then we need to decide what the value is going to be here. Something that might kind of help you to decide where it is if we look at the halfway point between 2 and 3, between 2 and 4, this has to be 3. And doing what we did earlier, well, it's a difference of 1 unit. So 1 unit is 5 boxes, and that means that it must be 0.2 for each box. And you can try that out and go up and see, oh, that does work, that does work. So we are 3 boxes up, so it would be 2.2 there, 2.4 and 2.6 units as our answer. And just make sure you're picking the right line to deal with. We're looking at the percentage decrease in lactate concentration at the end compared to the start at 52. So start, end. So let's deal with the start first of all, up to here. Run across to there. Each box is 0.2 as we said, so that's going to be 6.2, 6.4 at the start. And going up to, at the end of the season, it's at 4, bang on. So 4.0. One thing I check is, are we in the right order? Yes, we are, because it's decreasing, so it has to go down. Here, we're going to use the difference. So take them away from each other, gives you 2.4. Divided by the original, which was what we had at the start, which is 6.4. Times that by 100. Turn it into a percentage, 2.4 2.4 over 6.4 times 100 gives you 37.5%. Other ways of saying it is change over original times 100, decrease over original, decrease over start, whatever it is, but difference over original times 100 gets you your answer there. Calculate how many times greater the maximum lactate concentration is at the start compared to the end. So maximum is up here, so that's at the start is at 9. So just along there, and then this one's at 5. As I said, it's halfway between year 6 and 4, so it's 5. So it's 9 compared to 5. And we want to see, well, how many times bigger is 9? So 9 divided by 5 should give us an answer. So it's 1.8 times bigger. Now to prove that, well, if that's true, well then that times the smaller number gives you 9. That works. 9 is 1.8 times bigger than 5. For that would give us an answer there. Next one, different types of neurons. We have intermotor and sensory. Where is Y? So Y is taking in the information because arrows are going towards it. That must be then your sensory neuron. Here we've got it passing between ones. So this looks like it would have been the inter, not asked about. We've then got our motor neuron, which is carrying the information to the effector, which is in this case the muscle, which carries out a response. So motor would be in there. Synapse is a space between neurons. Electrical signals cannot pass through there, and they are carried by chemical messengers. Those messages will be chemicals going by, something like that. The effector produces a response. So as I said, this will be your muscle in this one, and they have a protective response from harm. It's preventing the cactus from harming your finger more than just a little jag. For that one, so that would be your answer there. They're protective, they're involuntary, so you don't need to think about them, and they're fast, or the reflex ones. Here we've got reproduction with sperm and egg. We're asked about what organ makes the eggs. That's the female, which in humans is going to be the ovary, produces the egg. What happens during fertilization? The nuclei, the bit with the genetic information of the sperm and egg, join. Fuse. Let's go for fuse. For that produces the zygote, which develops into the embryo. For that, unspecialized cells are stem cells. And specifically, these would be the embryonic cells for that one. And this one, we're talking about a condition that affects certain dogs, resulting in blindness. That's not a very cheery subject. Okay, so we're caused by the inheritance of a recessive form of the disease represented by R. And you've got an affected, unaffected, or a carrier. So they have kind of helped us out with the first bit. Okay, so one of the things that I find particularly helpful is if you write down all the combinations, so you can either have 2 dominant alleles, that's the big ones, you could have 1 of each, or you could have 2 of the little ones. Now the recessive one is the one that can cause the blindness and can affect the dog. So if they're affected, they will be baby R. So that means male 2 will be affected. They've already told us in here that this means they're called a carrier, which is not part of your course, but means they have the affected gene, but they themselves don't show it. And then 2 of the big letters means they are unaffected for that. And that means male 3 would be unaffected. Shuffling on, we're asked about different genotypes of them. So we have hetero and homozygous. Homo in science means the same, and hetero means different. So in this one, a carrier has one of each. That will be heterozygous for that one. Next one, the offspring were as follows: 4 unaffected and 4 carriers. Which male was chosen to breed with the female? That. Now, there might be a shortcut, but I think a good way to do it would just be to go through each of them. If this is the female, she has bigger, littler. Let's just try out the different combinations. So if we go for male 1, well, if they have one of each as well, we would end up with 2 big R's there, one of each, and 2 little ones. So in this case, they have carriers, affected and unaffected. That does not match the information that they're saying there, because they have all 3 of them, all 3 types. So it's not going to be that one. Let's try male 2. And no matter luck, it'll be male 3 will be the answer. So the female will still put bigger, littler, and then we have 2 littlers. So bigger, littler, bigger, littler, 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 littler. So we have affected and we have carriers, and it's a 50-50 split of them. But again, we've got no unaffected offspring for that. So must be male 3, but let's prove it. We've again got bigger, littler from the mother, bigger, bigger. So 2 big R's and R and R. So we've got half and half, 50-50, unaffected, sorry, and carriers, unaffected and carriers. What could be another way of doing that? Well, an unaffected one needs to have big R, bigger, and a carrier needs to have big R, littler. Well, if the mum has one of each, to ensure that all of them get a big R, the dad has to have 2 big R's. Because in that case, then all of them get a big R from their dad, half of them get a big R from their mum, so they're unaffected, half of them get a baby R, which means they'd be a carrier for that. So there are ways of doing it where you just kind of like work your way through it. Writing them down like the Punnett square way, that kind of shows it at a glance. So there's kind of pros and cons to all of them. Shouldn't take you too long to kind of write them out, but it does take longer than just try to puzzle it through in your head. In this one, we've got all their offspring were affected by it. Now, we've actually tried out all the different, well, not all the combinations. We've tried out a bunch of combinations. If any of the parents, so let's try this way with this one without doing a Punnett square. So if all the offspring are affected, then all the offspring must be little r, little r. If any of the parents had 1 big R or 2 big R's, you cannot make 2 little r's, because 1 comes from mum, 1 comes from dad. So that means the only way that could work is if both of the parents both were affected and both have only the recessive gene, the little r. And if you wanted to, oh, I'm gonna do it. You go through here, so you'll be able to see here. Well, all it is, every one of them are going to be little r for that one. And next one's asking about the different forms of the gene. That's what we're talking about, dominant and recessive. Big R, little r, they are different alleles, are the different forms of a gene that are passed on from parent to child. On to number 9, we've got a bit of text talking about COVID. Seems a long time ago and long COVID. So I'm going to skip through a little bit. We're talking about white blood cells. There are 2 types of white blood cells. There's phagocytes and lymphocytes. Now, it doesn't tell you in the paragraph what, like, information about what they are. Phagocytes engulf and digest. So it's like Pac-Man eating the pathogens, the microorganisms, and it's not that one. Lymphocytes produce antibodies. So in your pathway to getting this learned, they're the two options. Phagocytes engulf and destroy, lymphocytes produce antibodies. And that would give me my answer there. Moving on to the next one, we have to complete a table. And percentages at the start and the end. So let's go for a wee colour coding to hopefully help us work our way through it. So extreme breathlessness decreased from 38 to 30. Let's deal with that one first. 38, 30. Memory and concentration decreased 48 to 38. And our other health issues is there. That's alright. So 57 to 45 for that one. I was thinking, I was watching for them maybe saying it the other way around and saying something like back to front, and maybe, oh, at the end it was this, but it used to be that, to try and flip it around. This just pick the numbers out and put them in, which was a bit more straightforward than I thought at first. Next we want the whole number ratio about the different organs. So multiple, one, and no. So here they have done it in a different order. So it was 25 multiple, 65 1, and 10 no. So 25, 65, and 10. Checking that. 55, 65, and 1. Yep, so here they've done it the other way around. Now, when it comes to the ratio, we want to find a number that can divide into all of them. Well, 10 doesn't fit nicely into them all. 5 would. You can use your calculator. 5 divided by 5 just gives you 5. 65 divided by 5 will give you 13. And then 10 over 5 will give you 2. You have the calculator, so just use that. In some of them, you might want to go in like baby steps. So maybe like half it first, and then see what number goes into it. Make sure it's the simplest way it can go. So it's cancelled down as far as possible, and it's whole numbers, so no decimal places allowed. Give one reason why this study could be considered invalid. So this would mean there's a difference between them. What have we said here then? So other organs can be affected, other health issues. Oh, it's long COVID won't be the only thing they're dealing with. So they might have other health conditions as well as long COVID. Other things they're not telling you about is like their ages. So general health and this I've talked about. They are the mix between males and females, so the gender balance could be one of them. Humans are so complicated, there's going to be more than just one thing affecting that person's health. Here we're talking about the different parts of the heart. So we have got the atriums and ventricles. So atriums are like an attic, they're at the top. A attic, a atrium. We're at the bottom, meaning it's going to be a ventricle. When we look at that, that is on the left. So doctors obviously then decide that that's the right ventricle. That one, I do know it's because when they look at the patient, it's the patient's left and patient. They're just trying to confuse us. So if you look at it, if it's on the left as you look at it, as you've been taught since whenever you first learned left and right, we then say it's on the right. So it's the right ventricle, even though it's on the left. We are then asked about carbon dioxide concentration. Compare them in L versus N. So at the lungs, carbon dioxide is removed at the lungs. So that must mean it would be lower at L than N, as carbon dioxide is removed at lungs. Now, there'll still be carbon dioxide present in there. It's just going to be lower than at N for that. Hearts are supplied by the coronary artery, or there. Why would the heart cells stop contracting if it becomes blocked? That prevents supply of glucose and oxygen and respiration, they won't be able to produce ATP. And that'll mean there'll be less ATP for energy. It's lacking in supply of energy. Probably could have worded that better, but we'll move on. Exchange surfaces all have things in common. One will be a large surface area. That gets put in there. They'll also have thin walls to make it easier for the chemicals to pass through. There'd also need to be some sort of supply of maybe blood. Fish have blood, pretty sure fish have blood. And that carries away the chemicals and moves them away as well. This could be for the lungs or the villi for the food stuff as well. Large surface area, thin walls, both of them make it efficient exchanges of, in this case, gases. Gas exchange, yes. Draw a conclusion. Now, as the temperature increases, or as water temperature increases. So what does the question say, right? What's the effect of water temperature on the breathing rate? The average breathing rate is going up. So as water temperature increases, average breathing rate increases. So yeah, left hand goes up, right hand goes up. That sounds good to me. Next one, we're talking about distribution of organisms on a rocky shore. We have to calculate the average number of mussels per quadrat. Okay, so to get an average, we want to add all the values together and then divide by the number of numbers we added together. So it's just average, it's just quadrat, so that means we're going to want to have them all together. So 60, 58, 55, 50, 32, 30, 18, 13, 4, and 0. And then it's going to be whatever that total is divided by 10. So yes, the last one doesn't have any mussels, but you need to divide by 10 because it counts because it was tested. So it gives a total of 320 divided by 10 gives you 32 mussels on average. Just take your time, make sure you do the total at the top, and then divide it by 10. Sometimes the calculator can be a bit funky, but yeah, that would be an answer there. We increase the reliability. Well, we want to have more results, so we want to repeat the experiment for that. So we'll have more quadrats, and then or you could that would work as well as more sites to go further along, do more parts of the shore. That would do it as well. For that, have more of them, that would work. For that, next we're asked about water pollution and how different species can indicate / show what pollution levels are like. They are indicator species. Types of factors like pH that can affect organisms. They are non-living. They are abiotic factors. For that one, number 13, photosynthesis. Describe the relationship between carbon dioxide concentration and mass of sugar. Okay, so what we've got, two sections. So as carbon dioxide concentration increased to 0.08%, mass of sugar produced increased above 0.08%, mass of sugar stayed the same or stayed constant. At point X, it's not carbon dioxide stopping it because they've increased the concentration of carbon dioxide and it's not made a difference to the values. What else do plants need? Plants need light, so light intensity would be one of them. How much light there is. And again, with enzymes, photosynthesis is controlled by enzymes, so temperature has an effect on that as well. Hydrogen is a product of the light reactions is required for carbon fixation. How is hydrogen produced? Well, carbon dioxide plus water. Water is split into hydrogen and oxygen. Sugar produced and comes to starch. Name another one. Well, you can have cellulose. Makes plant cell. Oh, that would be the second thing. For that, this one, roll of pondweed and what happens to the energy at each level? Well, pondweed is at the beginning, it's a plant. So pondweed is a producer, which carries out photosynthesis to make sugar, glucose. So when it is consumed by a tadpole, around 10% of energy is passed on, and then the rest of it is lost. That's things like movement, heat, and waste materials. And then energy is lost at each of the next levels, meaning the pike has a lot less energy available for growth. That, and yeah, that would hopefully pick up all the marks there. For that one, next one, name a chemical and fertilizers used to help plant yields. Now, nitrogen, phosphorus, and potassium are the elements used in those. So I would just put that nitrogen or phosphorus or potassium. Next up, we have to do a bar graph. We need labels and scales. So the way a good graph should work is that the independent variable, the one that you're changing, should go along the bottom, and what we're measuring should go up and down. So the crop should go along the bottom. They've done that for us. How nice is that? So then we want to copy the other heading, which is average loss in yield, and that will be in percent. We next need to look at what numbers would be good to go up in. Now, they've kind of helped us out by telling us that the insects are at 4 for that. So that would suggest that we are starting at 0, and every big box would be 2. So 6, then 8, which would mean this one, halfway between the two, which would be 7, and it is, then 8 and 8. And that looks good to me. We need to go up, because 14's our highest. So 12, 14, might as well finish it off with a 16. So then we just need to then put in our numbers. And that would pick up both the marks for there. A couple of things to remember: if you're in charge of more of that graph, whatever you're changing goes along the bottom, your measurements, your dependent variable goes up and down the way, your scales have to be even, and you want to be taking up at least half of the graph paper with your information. So we've ticked all those boxes. One last double check: 12 for that, and 8 for the oilseed. Oats are 14 and 8, yes. 6 and 7, yes. 7 and 4. And that looks good to me. So we want the lowest combined percentage loss from the two causes. We want to just add the two of them together. So we get 11, 13, 22 and 20. Lowest would be wheat. Wheat would be our answer. So it's incorrect to say that each crop is more diseased than insect. Well, which one has it the other way around? Barley is more affected by insects. So it's the other way around. Barley more affected by insects. Total crop harvested was 140,000 tons. What would have been produced if it had not been affected by the plants? Okay, so oilseed was 20% lower. We had 20% loss. Okay, so that means then that what we've got is 80%. So the stuff that wasn't affected is 140,000. I would probably do here is, well, if we lower it down to what 1% would be, so 80 divided by 80 gives you 1. Calculator you can use, like, if you need to figure out how it all fits together. And then how do we go from 1 up to 100? We can multiply by 100. So then if we do the same on the other side, 140,000 over 80 times 100 into the calculator, and then I get 175,000 tons as an answer. That's my kind of way I would deal with them. The build-up of pesticides in the cells, we're accumulating, it's bioaccumulation. And that's why the ones at the top of the food chain are more affected by the chemicals than the weird ones often. It builds up in their system. Editor, especially using alternative. We're using animals, a living organism to control the numbers. Biological control. Followed by the next question, which is talking about types of birds that have evolved. They must be split by an isolation barrier. So what kind of things can keep them apart? We could have a geographical barrier. The other ones are ecological, something in their environment, or a behavioural. Or that one. Name the molecule within DNA in which mutations would occur. Mutations are random changes to DNA. The genetic information, so DNA is the one for that. You can have irradiation. So if people are exposed to high levels of radiation, then that can have an effect. You can have chemicals. Would be things that can increase the rates of those. Suggest how selection pressure could have led to the differences in the honey creeper. So their beaks are different. Food, eating different food. They what would the selection pressure be? So available or competition. So if they couldn't get access to one particular type of food. Okay, this one for a species. They are a group of organisms who can interbreed to produce fertile offspring. That brings us to the end of the paper. Thanks for watching. Goodbye.